Hey guys, we're gonna come back and do this last part of photosynthesis, what I call photosynthesis in a nutshell. Um, if you read the chapter, you know that there's a lot of information there, and we're just doing mainly these highlights, and this is what you need to know. So let's get into the first reaction, which is the light reaction or the light dependent reaction. Here we know that chlorophyll, which is one of the pigments that we're just highlighting, it absorbs solar energy and energizes electrons, and we know that those electrons pass down the electron transport chain. And because those electrons pass down the electron transport chain, we know that water will be oxidized or water will be split. And if water is split, we know that the product of this light reaction is going to be oxygen gas. So we know that the substrate for this reaction is water. The product for this reaction will be oxygen gas. One of the other things that we will find out is that solar energy is going to provide energy so that we can create ATP, because as those electrons are being passed down the electron transport chain and those hydrogen ions are being pumped across the membrane, we know that that's a part of what we call chemiosmosis. I'll get into it when we get into cellular respiration in a little bit more detail. But we know that we're going to store some energy in the form of ATP and NADPH. So in other words, we know that ADP plus P is coming into this thylakoid membrane where this reaction is taking place. We also know that NADP plus is also coming into this membrane as well. So NADP plus gets reduced to NADPH because it's picked up electron and hydrogen ion. We know that ATP, ADP was created from ATP plus P. All right. So this is showing you that thylakoid organization. And here we see water being oxidized. Here it says we've created a half O2. The electrons are being picked up. They're being passed from one carrier over to the next carrier, down to the next carrier. And we see that NADP plus picks up a hydrogen ion, also an electron, and becomes NADPH. The hydrogen ions, we know that they are being pumped into this thylakoid space. And it says as these hydrogen ions pass through the ATP synthase complex, we can take ADP plus P and synthesize ATP. Okay, and it's simply from the flow of these hydrogen ions from an area of high concentration to an area of low concentration. But again, we'll get into the specifics of this process in the next set of videos. All right. So what are Calvin cycle reactions? What do they do? So remember, Calvin cycles, not only the Calvin cycle, we also call it the dark reaction. We call it the light independent reaction. Well, this is a series of reactions that are responsible for creating or producing carbohydrate molecules. If you remember the formula, it said that if we took in water from the roots and carbon dioxide from the air, or so it would be atmospheric carbon dioxide. It said at the end of this reaction, not only would we have oxygen, but we would have glucose. What we're going to see is that glucose is not the final product of the Calvin cycle reactions, but instead the final product of Calvin cycle reactions are the pGal or G3P molecules. And so that's what we're getting ready to get into. All right, so it says The reaction number 2 is a Calvin cycle reaction. It says in this reaction, what we're going to see in the next slide is that carbon dioxide is going to be taken up through the stomata and then fixed and reduced to a carbohydrate. So we said step 1 is that fixation process. So in the fixation process, we know carbon dioxide will have to be fixed to a molecule that we're going to call RUBP, okay? So one of the things about RUBP is actually ribulose 1,5-biphosphate. It is a 5-carbon molecule, okay, that has a phosphate group in the first position and a phosphate group in the 5th position, okay? And it says we're going to take this 1-carbon molecule called carbon dioxide and we're going to fix it to this 5-carbon molecule that's called RUBP, and that's going to result in this 6-carbon molecule that's unstable that we're going to call PGA, okay? And so we know that this process, again, is going to utilize atmospheric carbon dioxide. So again, after reduction, the final product for the Calvin cycle will be G3P or PGAL. I actually say that G3P is the molecule that you get after the splitting, but it's actually PGA. And then after a series of other reactions, it becomes G3P or PGAL. And what we're going to find out is that G3P or PGAL is very important. So we know that the reduction actually is what's producing us this G3P or PGAL. It says what enzyme is responsible for covalently bonding CO2 to RUBP, which is the ribulose 1,5-biphosphate. That enzyme is called Rubisco. We know that enzymes help lower the activation energy and help speed up these reactions. So again, if you remember from chapter 6, it's not that these reactions won't take place on their own, but enzymes help to speed up these reactions. It helps them to align themselves in the right position so that the bonds can take place. All right, so, and then we said step 2 was the carbon dioxide reduction, and we know that the ATP is important in this reduction process, and the NADPH actually releasing those hydrogen ions is part of this reduction process in order to produce the G3P. So let's talk about G3P. Oh, well, before I get into G3P, let's go back and highlight this. So it says, you remember in our light reaction, we said what was the molecule, what was the substrate that was being used. We said water was the substrate. It went into the thylakoid membrane. Okay, and the thylakoid membrane is that chlorophyll complex that's going to absorb this solar energy and excite the electrons. It says when the electrons are excited, they get passed down the electron transport system. We know that water is being oxidized or split. So when water is oxidized or split, we said the product of that reaction would be oxygen gas. Okay, so oxygen gas for this reaction. So we just finished the Calvin cycle reaction, so we know that carbon dioxide is the reactant for the light independent or dark reaction or Calvin cycle reaction. So we know that it is moving into not the thylakoid, but the stroma. So this fluid filled space here is called the stroma. So we know this guy moves into the Calvin cycle. We know that Rubisco helps speed the bonding of carbon dioxide to RUBP. And we said that this 6-carbon molecule now creates uh, uh, an unstable intermediate that we're going to call PGA. So we know that there's 2 of these because this guy gets split in half. So we know it's actually 2 PGAs, you know. But anyway, after reduction and other reactions, we know that the product that's going to actually come out of the Calvin cycle is pGal or G3P. And I probably should have written G3P and I would have had less to write. So both the same entity, but we can call it pGal or G3P. And then it says at the final end of this reaction, it says pGal is important or G3P is important because it says this is where we're going to get the glucose from or other organic molecules. So it says if we take 2 times pGal, it says we're going to get glucose. So glucose, which is what you see coming at the tail end of that reaction, is glucose, which is C6H12O6. It says, but it says pGal is important because pGal, the backbone of pGal can be used to make a host of other organic molecules. Okay. All right, so again, the importance of the Calvin cycle, again, it says that G3P or pGal is the product of the Calvin cycle that can be converted to a variety of organic molecules. So again, it's showing us the carbon dioxide coming in. We end up splitting into this PGA after it's been reduced so we can use the ATP phosphorylated. It says, and then we use the NADPH. Now that's where we're getting the pGal from. And which is what I just mentioned. It says 2 times the pGals. That's how we produce the glucose. And then it says, but pGal, if we go in the opposite direction, it says will produce a numerous amount of other organic molecules. All right, and this is just showing us that pGal is very important because the backbone of pGal, the structure can be made to make fatty acids, amino acids, fructose phosphate. It says if we take fructose phosphate, it can create glucose phosphate, which will create sucrose, starch, and cellulose. So let's go on to this next slide. And this is just giving you some highlights from that previous slide. It says pGal can be used for synthesis of starch and cellulose. We know that starch is the storage form of glucose. We know that cellulose is a structural form of glucose. We know that us animals cannot digest cellulose, nor can we make cellulose, so it's used for the fiber in our diet. So starch again is a storage form of glucose. We said cellulose was a structural component. If you remember from chapter 1, it said that cellulose used beta glucose, and we do not possess an enzyme that can break those linkages. Therefore, it becomes the fiber in our diet. Okay, so that again, those linkages make it indigestible to us. We don't make an enzyme that can actually digest that glycosidic bond. All right, A plant can also utilize the hydrocarbon skeleton or backbone of pGal to form fatty acids, and we know that fatty acids and glycerol, and we know that fatty acids and glycerol are two of the main components that we need to make up oils. So whether it's corn oil, safflower oil, olive oil, we know that we would need 3 fatty acids and one of those glycerol molecules in order to make up the oils. And that takes you all the way back to chapter 1. Okay. And then it says if we add nitrogen, it says we can use this hydrocarbon skeleton of pGal and make amino acids. So that just kind of gives you a basic overview of photosynthesis. I hope that you guys have had a wonderful overview. And if you have any questions, you can always reach out to me. There's a study guide and questions usually following the videos. Have a wonderful day.